So 2 a.m. over in California. We're very brings us to the next speaker, Dr. Priya Shah from the University of California, Davis. Priya, thank you very much for joining us so early in your day. And I guess the floor is yours. Handing over to yourself.
Thank you very much, Kamran, for your invitation and for the introduction. I'm really excited to be here, even though it is pretty early in the morning. So let me just find my slides here.
Yeah, we can see them.
All right, great. So my group, very similar to Kevin's group, is focused on these molecular interactions for dengue virus. And what we're specifically interested in is these molecular interactions between the virus and host proteins that might reveal interesting aspects of virus replication and disease. And so what I'm going to tell you about today is some of our work, but how that might relate to identifying novel antiviral therapeutic strategies. And I'll give you one example of some of the work that we've done in this area. Okay, and so I think Nilika really nicely outlined the dynamics of dengue disease and how viremia happens early along with the fever, and then you get the severe disease following on that. And so both Nilika and Kevin's group have done some really exciting work related to treating that disease, but also diagnosing that severe disease. And what I want to emphasize is that what I'm going to be talking about in this short talk is dealing with some of the molecular level events that happen during that viremia phase, that virus replication phase, that could be targeted therapeutically. Okay. And so when we talk about these types of antiviral compounds that actually target the virus and inhibit virus replication, typically these are small molecule compounds that target a viral protein. And so I'm giving you one of the exciting new examples of these antiviral compounds for dengue virus in particular that was not published by my group. So this is work done by some very talented scientists that was published in Science Translational Medicine earlier this year, some very eminent dengue virologists and clinicians. And what they identified was this chemical compound called NITD 688 that targets a dengue virus protein called NS4A, or non-structural protein 4A. And that is shown right here. And it's shown right here in this cartoon. And so this chemical compound, it serves to inhibit the function of this viral protein, and that's how virus replication and viremia are lowered. Now, the issue with a lot of antiviral therapies that target viral proteins is this problem of drug resistance. And we've seen this through the early 90s with HIV until the combination therapies were developed. We have been worried about this with our influenza therapies that are very useful right now. And we've also been worried about this with SARS-CoV-2 and the emergence of not only variants that are more pathogenic, but also variants that evade some of our great tools in the clinic, like remdesivir. And so what you can see in this diagram here are 3 mutations that are indicated by these stars here that allow this dengue virus to evade or escape inhibition by the small molecule compound. And so these viruses, you know, they're incredibly effective at evolving to evade any sort of selective pressure. And so viruses, especially RNA viruses, are going to rapidly evolve capabilities like this. Now, these viruses do exhibit lower replication capacities. They're not as capable of replicating as the wild type or original isolate in many cases, but viruses can always then continue to adapt and improve upon those escape mutations, right? And so this is one of the concerns that we always have in developing antiviral therapies. And so what I want to talk about today is a different approach to developing antiviral therapies, where we target not the virus, but the host. Okay, and the reason we're going to do this is because all viruses have to interact with and hijack host cellular proteins to replicate. So now what I want you to do, we've been talking about, this sort of organismal and tissue level disease. What I want to do is zoom down to the molecular level and talk about what's actually going on inside of a single cell for dengue virus to replicate. And if you think about it, any virus, including dengue virus, it has to do three things. It has to get inside the cell, it has to replicate its genome, and then it has to get out of the cell, okay? And to do all of these tasks, viruses interact with host proteins through physical interactions, which we depict through these cartoon diagrams where the square in the center is a viral protein, and all of these circles are human proteins that viral protein physically interacts with, is connected to in a very physical sense to facilitate these very basic tasks of virus replication that have to happen inside a host cell. And so with dengue virus, because it's a mosquito-borne virus, these basic aspects of virus replication, they have to occur in both the human host and the human cells and mosquito cells. And so one way of targeting the virus where it really hurts is to identify these virus host protein interactions that are essential for replication in human and mosquito cells and go after those essential features to inhibit the virus. And so that's exactly what we've done with dengue virus host protein interactions. And so what we did is we identified these virus host protein interactions that I'm showing as a cartoon here for every single dengue virus protein in both human and mosquito cells. And we did this by purifying the viral protein in either human or mosquito cells, and then analyzing all of the host proteins using mass spectrometry. Okay, and so I'm glossing over many years of work when I show you the cartoons here that summarize the data. And what you're looking at here in the blue are all of the dengue virus human protein interactions that we identified, that we found to be the most significant. And there are about 200 of those. And what you see in the gold on the right side of the slide is all of the dengue virus mosquito protein interactions that we identified to be high confidence. And there's about 150 of them that we identified. And so what we can do is we can use a lot of bioinformatic network integration approaches to basically combine these two networks and identify what's shared between the two. And we can do this at 3 different levels. We can do this with individual proteins. We can do this with entire protein complexes. And then we can do this with biological pathways and processes in the cell to see what's the same and what's different. And so that's what I'm showing you here in this slide. And so if you focus on the diagram on the left side of the slide, what you're seeing is that integration of all the protein interaction network data for the human proteins, which are in blue, and the mosquito proteins, which are in this orange color. And so here I'm showing you data for just one viral protein called non-structural protein 4A. And I'm showing you this data in particular because it was really exciting to us. What we found were that, first of all, this protein had a lot of shared characteristics in the human and mosquito cells in terms of the types of proteins it interacted with. It interacted with host proteins that were involved in oxidative phosphorylation. So that's, our aerobic respiration cycle that eukaryotes take advantage of. We saw a lot of proteins involved in organelle targeting and organization, in glycosylation, because as we know with proteins like NS1, they need to be glycosylated to get out of the cell. And we also saw a lot of proteins involved in trafficking and potentially involved in that entry and exit of the virus from the cells. And so that's at the pathway level. We could also zoom in on individual proteins. And that's what I'm showing you in this panel here, where you can see the individual proteins that were either identified in the human cells or the mosquito cells that make up this category involved in organelle targeting and organization. And the proteins that I want to highlight to you are part of this complex called the Sec61 translocon. This is an essential complex found in all eukaryotes that is involved in transmembrane protein biosynthesis. And so dengue virus, like all other flaviviruses, have a lot of transmembrane proteins. And so taking advantage of the Sec61 translocon will help the virus translate and produce functional transmembrane proteins. And so what we decided to do was collaborate with a professor at UCSF, Jack Taunton, who specializes in studying this translocon, the Sec61 translocon, and the fundamental biology behind it. But what they've also done in the Taunton group is they've developed small molecule inhibitors of this complex. And so we took these small molecule inhibitors and used them to inhibit dengue virus replication in human and mosquito cells. And so here you can see what the normal virus replication looks like in both of these cell types. And when we add either inhibitor for the Sec61 translocon, that virus replication is essentially undetectable using our plaque assay methods. Importantly, one thing that you might be worried about, especially when you target a human complex, is toxicity to the host. What's exciting about these small molecule inhibitors is that they really modulate the activity of the translocon. They don't inhibit it outright. And so this complex still has the ability to synthesize many transmembrane proteins, including the ones that are essential for cell viability. It's just a select number of transmembrane proteins that are affected by these inhibitors, including flavivirus proteins. And so what you can see here is a measurement of cell viability for our DMSO dimethyl sulfoxide carrier control, along with the two inhibitors. And the cell viability is essentially unaffected by these inhibitors. What else is exciting is that we can do time of addition assays to show that even when we infect before we add the drug, if we add the drug at later points, we can still inhibit virus replication, which is what you see here with protein synthesis of NS2B, another viral protein that we can use to track virus replication at very early stages. And so this is one example of how these conserved interactions with host proteins have a really strong therapeutic potential for dengue virus, but also other emerging diseases or neglected tropical diseases as well. And what this can do is help treat patients in that viremia phase to help get them back into the workforce if they're simply suffering from dengue fever, because it's still a very debilitating disease, or even help prevent them from producing a more severe disease like dengue hemorrhagic fever and shock syndrome. Okay, and so with that, I'd like to thank the members of my group at UC Davis and also my collaborators, past and present, especially Nevin Krogan, my postdoc advisor, who I did a lot of this work with, and Jack Taunton, who created those Sec61 translocon inhibitors, and my funding sources. And of course, I'm happy to take any questions. And I think there's going to be a great panel discussion at the end of this.
So thank you very, very much for that, Priya. Fantastic and very complex subject area. I love the mapping of human interaction, the protein and the mosquito interactions to yield potentially new targets. Fascinating approach, and hats off to you and your team, your lab for running that.